Can you see my screen?
Yes, we can.
And I will go full screen mode. So yeah, what I'd like to do today is to introduce the Global Alliance for Genomics and Health, GA4GH Phenopacket schema, and show you some use cases in rare disease, long COVID, and oncology. But before I do that, I'd just like to give an introduction to ontologies, which are one of the essential building blocks of the schema, and to show how semantic or ontology algorithms can be used to assist in the diagnostic yield in rare disease. So what is an ontology? Well, ontologies actually go back to Aristotle. If you look at Aristotle's physics book, which basically stood for nature, there are many hierarchical descriptions of animals and other things in nature that resemble modern ontologies. But actually, in our sense, ontology is a form of knowledge representation, which is one of the pillars of artificial intelligence. Of how the various ontologies that the open source community have developed over the last decades are now coming together to enable sort of a comprehensive model of disease. And so just to go through this example, we're talking about Fanconi anemia. And so Fanconi anemia is a clinical diagnosis that corresponds to about 22 genetic subtypes. So those are denoted by ontology terms. They're caused by variants in specific genes, which can be represented in ontologies. And these genes are members of a pathway that can be described by several tens of gene ontology function terms. What our group developed in Berlin and continuing to develop here in the US is the human phenotype ontology, the HPO. Those are the yellow ones in the middle. I don't know if you're actually seeing my mouse, but for instance, anemia, thrombocytopenia. In actuality, Fanconi anemia has, on the order of 70 phenotypic features. So this is just a subset. And we feel that we can compare not only patient profile to this disease model, but we can also compare different diseases to each other to learn about pathomechanisms and perhaps to pave the way for precision stratification. And so in this example, acquired aplastic anemia shares the hematological feature, or many of the features with Fanconi, but doesn't share some of the other items such as microcephaly. And so let's see. This is the HPO. There are roughly 17,000 terms. Each term describes a specific phenotypic abnormality, a specific phenotypic feature. And there are a few things that distinguish HPO from other terminologies in use in medicine, such as SNOMED or many others. Firstly, there's no other resource that has so much detail about the signs and symptoms of human disease. And in contrast to other terminologies that were developed for purposes such as running a hospital or billing, the HPO was designed for scientific computation, for sophisticated algorithmics. And one example is that the terms are defined on the basis of OWL class definitions. I'll skip the details, but basically this is a logical definition of the terms with reference to other ontologies for anatomy, biochemistry, histology, and a few others. There are also nearly 160,000 annotations. This figure is a bit old. I'm sorry, even more than that for monogenic chromosomal diseases. I showed you all of the papers here because a lot of these represent community contributions. So we've been ontologies, databases will have trouble understanding. But the the interesting thing, or let me continue along the line of how, I told you about the computational definitions. Each class is also defined with an English language definition, with synonyms that are useful for text mining, and cross-references to other resources that are also useful for mapping data from electronic health records. The scope is really any of the manifestations of diseases. So those of you who are physicians will know that there are some diseases that you can diagnose based on their gestalt, so the overall picture. For instance, if you see a child with Down syndrome, probably most of us would just recognize the diagnosis, but this doesn't scale to the roughly 10,000 known rare diseases and roughly 10,000 common diseases. So what physicians do is to write down the individual signs and symptoms, lab findings, results from X-rays and other imaging studies, electrophysiology, many other things, and to use computer programs or just to look it up in a book to try to find the diagnosis. This is known as deep phenotyping, and that's what the HPO is designed to support. So when I was in Berlin and I did my training as a pediatrician, and I decided to go to into genetics, also at the Charité, and I found it was a very difficult challenge to be able to recognize the many rare diseases based on their gestalt. And so I also turned to one of the existing computer databases there, and that basically, you would enter two terms. There's roughly 1,000 terms in the London Dysmorphology database. You would enter two terms and you would get an unordered list of 300 candidates that share those two terms. Then you would enter a third one and you would get zero. There would be no disease with all three terms. And this seemed like basically it's not an intelligent way to do this search. And so this was really the inspiration to try to come up with the HPO to enable a specificity weighted search such that you would get an ordered rank of diagnoses, but also a fuzzy search such that if the terms you enter weren't quite right, you would still get some match. You would still get a reasonable list. And so the way we came up of doing this was to first define the specificity value of each term based on the information content. And essentially what this equation says is the rarer a term is in your database, the higher the information content. And so our database now has roughly 8,000, let's just say it has 10,000 diseases. And so if one of those diseases has a certain HPO term, the frequency of that term is 1 in 10,000. If you take the negative logarithm of that, you get an information content of 4. If 10 of these diseases have that, the frequency is 1 in 1,000 and your information content is 3. There are mathematical reasons why this is a good way to represent the amount of information you get from a certain observation. And just to explain how we use this in algorithms, let's look at this example ontology. So this is about carnivores. And so we have, let's say, a library of 1,000 books about carnivores. Some of these are about dogs and some of these are about cats, canine and feline. And it goes down more and more detailed. So this is essentially the same thing as the HPO database, except that there are HPO terms instead of animals. And so the information content of, for instance, beagle is defined by 40 divided by 1,000. And the thing is that all dogs are beagles. I'm sorry, all beagles are dogs. So all of these 40 annotations basically get included in the count of dogs. And so if we add up, we have 40 beagles, 5 terriers, 15 huskies, that's 60. But there must be 200 and other books that are just about dogs in general. So this is an important thing to realize. This is why the specificity goes up the further down the ontology you go. And so if we want to measure the similarity between cheetah and lion, we go to their most specific common ancestor, which is big cat, and we take its information content, which is 1.3 in this example. In contrast, if we want to know how similar are beagle and wildcat, well, they have no common ancestor outside of the root. Since all terms are, since all books are about carnivores, the frequency is 1,000 divided by 1,000. And so the logarithm of 1 is 0. So we get an information content of 0. So there's no similarity. And to turn this into a clinical tool, patients don't just have one HPO term, they have many. And there are a few ways of putting this together. But essentially what this does is you look for the best match for each term, and you take the average of that match. So that's how semantic fuzzy specificity weighted matching works. And I'm just going to skip that. So if we're looking for how to use this today in the age of genomic medicine, well, many of our patients will come with an exome or a genome sequencing. And, in Berlin, my department head, Stefan Mundlos, got, I believe, the first Illumina machine in Europe at late 2009. And we sequenced a family, and at the time there was no software to analyze exomes, and it took us 3 months to go through each of the variants one by one in essentially an Excel file and Google. And after 3 months, Peter Krawitz, who is now in Bonn and was in my group at the time, said, "Oh, this is an obvious candidate for various reasons." And today we could have done the same procedure in about a half an hour, just using software that people have developed in the last 13 years. But the baseline is 3 months. So that's what software is trying to help. And there are a number of things, but one of the things that you can do with HPO is to assess the phenotypic relevance and to rank the candidates based on how, so you rank variants that are predicted to be pathogenic based on how well the phenotype matches. And so to do that, each of the diseases in our database has a disease model that consists of a list of HPO terms. So this is a disease called familial cold autoinflammatory syndrome. Each of these annotations, as we call them, can have metadata such as the frequency. So for instance, 3 out of 3 people in a certain PubMed study were found to have episodic fever. The model includes other things such as medical actions. That is a new ontology that we're just finishing this year. And for instance, the administration of this medication will prevent episodic fever. And this is something that we think will add to the computational model in an interesting way. So I told you about fuzzy specificity weighted matching. So essentially, these were patients we saw in Berlin. The blue terms are essentially the database version of a disease called Wiedemann-Steiner syndrome. And but the patients, they matched in many ways, but a lot of them were fuzzy matches. For instance, the boy had a big skull while the girl and the database have a small skull. And using our procedures, these got ranked on ranks 1 and 2. We've implemented this software in these algorithms in a software called Exomizer that has been used in many places. I think probably the best advertisement for this was the 100,000 Genomes Project. And they basically wrote that about 50% of the diagnoses were helped by use of Exomizer and HPO. So I'm going to turn now to Phenopackets. One of the premier use cases of Phenopackets is to support the kind of differential diagnosis that with HPO and with Exomizer and other tools. These tools now, not just from our lab, but some of the other tools that are out there, commercial and academic, are starting to support the Phenopacket format. This makes it essentially easier to use these tools and also to compare different tools. But patient phenotyping, and this is something which, in many ways, is still sort of 19th century technology. This is how I learned to phenotype. You just write things down. And I told you about our experiences in 2010 without software that used things like VCF. But standards enable computation. Standards would have accelerated our gene discovery back then from 3 months to maybe a few hours. And there haven't really been standards like that in general to represent individual patient courses, especially not for rare disease. And so that was our goal, was to make a standard that would be similarly influential as VCF and other standards are for genomics. And so we joined the Global Alliance for Genomics and Health. We've been working on the Phenopackets, well, it's finished, but we worked on it for a total of 7 years. So there were literally hundreds of conferences and Zooms and live meetings, and a ton of people gave input into developing the standard. This is an overview of the standard. Happy to answer some questions about the technologies we use to generate this. It involves something called protobuf, but you can think of this as a JSON schema, a hierarchical schema of information. And in the following slides, I'll be presenting a little bit more detail, for instance, about the phenotypic feature, which is here, the measurement, and a few other things. But essentially, a phenopacket represents data about one patient over time. The patient can have one or more disease diagnoses. The patient can have phenotypic features. These are HPO terms with context, as I'll show you. The patients can have measurements. These are quantitative measurements, such as a platelet count. There can be medical actions, treatments, procedures, and there's an interpretation element that uses another GA4-GH standard called VRS. So, and what I'm going to do is I'm going to present an example case that we published here. This is about retinoblastoma, and this shows off most of the elements. So if you're interested in learning about Phenopackets, this article is probably the best place to go. And so each of the elements is defined using protobuf. That's what we see in the left. I'm going to skip the technical details. But essentially, this is a hierarchical representation of an ontology class, in this case, which has an ID and a label. And so this is a YAML representation. This is a JSON representation. So I'm just going to go through some of the elements. So a phenopacket will start off with some demographic information. There are various ways of using this to protect privacy. So for instance, we can represent the date using the age, using this ISO 8601 duration. This means 6 months. We can also use an age range if we want to protect privacy. So, since we developed the HPO, we've gotten tons of emails like, can you help me? Here's a list of HPO terms. Sometimes people would send it with Excel, sometimes with Word, whatever. Always different. And this is now a feature of actually saying, don't just send me the HPO term, give me a little bit more context. And so phenotypic feature includes both an ontology term as well as fields, optional fields for severity, modifiers such as quotidian, which means once a day, the age of onset. So in this case, infantile spasms, type of epilepsy, had onset at in the first year of life, infantile, and a resolution, so it went away at age 4 years and 2 months. So this is something you see not uncommonly. The same child was found not to have global developmental delay. So this was investigated but excluded clinically. This is how it would look in a computer file. And so a phenopacket will have a list of phenotypic features. These are the features that were found in our patient with retinoblastoma. And so this basically means your eye is white if you shine light on it, and there was retinal detachment, and so on. Measurements, so an HPO term is essentially yes or no, but sometimes you actually want to keep the original measurement. And so in this case, platelets were found to be 24,000 cells per microliter. That's low. So if you wanted to use an HPO term, it would be thrombocytopenia. But there are many situations where you would actually want to keep the original measurements. And so that's what you can do with this. I'm going to skip that detail. Medical action. So roughly speaking, this is treatment, but actually not just treatment. There are many medical actions that involve, for instance, giving patients advice to avoid a certain type of food or sunlight. That's something you might see with rare disease. But this is an example of a patient who had COVID, who in 2016 had heart failure, got a left ventricular assist, and who was admitted in 2020 at the beginning of the pandemic. Initially was given oxygen by nasal cannula, 2 liters a minute. Over this period of time, the patient got very ill and was then given 50 liters a minute for a day or so. The patient was also treated with the Regeneron antibody cocktail, and this is sort of twice a day, a certain number of milligrams, and this and this was the total interval while it was used. So this is the sort of thing that you can now represent and also compute over, and this is what it would look like as a computer file. Another thing that's important for many types of medicine are biosamples. So biopsies, or in the case of our patient with retinoblastoma, the tumor was, the entire eye was removed. And so you can have phenotypic features of that tumor. So for instance, rosette formation, this is something you see underneath the microscope. And there was also apoptosis and necrosis, something you see underneath the microscope. There was a measurement in this case that was the size of the tumor, so 15 millimeters. And there was an assessment of the pathological TNM stages, and the procedure by which this biosample was obtained was enucleation, so removing the left eye. And this was then sequenced, so there was a genome sequencing not only of the blood, but also of the tumor sample. And this then can be used to give context to genetic findings. So in this instance, there was a nonsense mutation that was found in the biosample in the tumor. And this is the second half of that assessment of the biosample. There are many more details in the article that I showed you at the beginning. I was just trying to give you a flavor for the kinds of information that can be captured. But so Phenopackets can be created from a number of input sources, including electronic health records, applications. We have software now to essentially transform OMAP, which is a research database for EHR data. I'll show you a little bit about the FHIR version of this. We have various applications that are available now in prototype on the web, by which you can use text mining and other tools to enter accurate and comprehensive phenopackets. We think that if you sort of take very disparate data and code it as phenopackets, then you can use the same set of software to analyze data that's coming from many different sources. And so we think this is useful for diagnostics. So tools such as Exomizer take phenopackets as input, but you can also use this in an EHR to identify cohorts. This will be supporting the version 2 of the G4GH Matchmaker Exchange. We can use this for translational research. I'll show you an example on long COVID in a second. We are talking with journals in human genetics and have been told by basically all of the 10 journals we spoke with that they would be happy to ask authors to submit phenopackets together with articles, such that with time we will build up a database of individual case reports that would be computable. And consortiums such as the SolveRD, a European consortium that I've been involved in, are using Phenopackets to exchange data. And we've had roughly 15,000 cases coded in this way. And that makes it easy to set up pipelines to analyze that number of cases computationally. I'm not going to talk too much about FHIR because there's a lot to say, but essentially we've created a version of Phenopackets in FHIR for use in electronic health record systems. We are a part of an HL7 project called Vulkan Accelerator and are attending the so-called HL7 Connectathon meetings to get input from other FHIR groups. And we're planning to bring this forward to balloting to make this an official HL7 project, but that takes several years. So we're kind of in the middle of that process. The Phenopacket schema is free and open source. So there are the top link here is the place to go to see it. This is also linked to detailed documentation. There are also a series of tools. So these are the FHIR IG. Phenopacket tools is a software library that will help you to build Phenopackets with Java. There's a similar library that we've written but not going to publish that does this in Python. You can feel free to get in contact with me. We got approval from the International Standards Organization, and if you want, you can pay 100 francs to get it, but it's identical with the open source version. And this was a special deal with ISO because who are now trying to support genomics more. Do I have time or sorry, I don't see the clock.
You have another 10 minutes, so you can go.
Okay, I'm gonna maybe go. Okay, I think I'll have time. So the use of Phenopackets is not just limited to rare disease. We think it can be used in many contexts. And so one of the things we're doing is to try to encode data about patients with COVID or long COVID. This is just beginning. And we're essentially developing a tool to translate from OMAP to Phenopackets to capture the clinical course. And to do this, we first did a lot of curation. Basically, we read a ton of articles about long COVID and identified 200 manifestations that had been reported for long COVID and developed HPO terms for them. And this is the kind of thing that you can now do with this. So we're involved in an NIH project called the National COVID Cohort Collaboratory. We have millions of patients with COVID-19. Now this number is closer to about 9 million patients with COVID-19. So all of this is from 70 hospitals in the US. And recently, the ICD came up with a code for long COVID called U09.9. And we can use this to reliably identify patients with long COVID. And our question was, and this was motivated by, many other studies and just talking with patients. And, some patients with long COVID have very serious manifestations in many different organs, but many patients sort of just have pulmonary or cardiological or whatever symptoms. And we wanted to know, can we actually detect that using EHR data? And so I explained the fuzzy specificity weighted algorithms. What I explained before was compare a patient to a disease model. You can do something very similar to compare two patients. And essentially, if two patients share a lot of HPO terms or are close to each other in this hierarchy, they're highly similar to each other. If they don't share many terms, they're not as similar to each other. And so we can basically create a similarity matrix and cluster that with a very simple method. And we did that and found 6 clusters. And we were able to show that some of the clusters were very serious and that patients tended to have manifestations in multiple organ systems. You probably can't quite read this, but the rows are pulmonary, neuro lab, immunology, and so on. And this gives maybe a slightly easier to read version of that. So cluster 1 was enriched in several lab findings that turn out to be, in other studies, to be associated with severe COVID. And there's also a relatively high frequency of manifestations in many other organ systems. But for instance, this cluster 2 is enriched in what we call constitutional symptoms, pain and fatigue. Other clusters are more enriched in things like cardiology and down here. So, and we could actually show that the clusters that we defined in data partner 1, basically hospital 1, generalized to 5 other hospitals that we tested with, and essentially we never saw the same degree of similarity. So we could essentially get z-scores that would be closer to 1,000 if we continued the permutations. And when we looked at other sort of covariates, for instance, mortality that we hadn't used for clustering, we also saw that there was some degree of generalizability in that the cluster 1 showed the highest mortality in all hospitals. So that's so I'm gonna stop there and maybe take questions. Just mention some work that we're now planning in cancer. So we've been talking with NCI, who basically have 20 different databases in 20 different departments, such as leukemia, lung cancer, colon cancer, and each of these databases is different. And they were all developed to a specific purpose, but they were developed 20 years ago and have different schemas. And so it's very hard for NCI to put all of that data in one place. And so what we are going to be doing is essentially to create a transformation function, so-called extract, transform, load function, that will transform the data from these databases into a common model of Phenopacket. And then it is possible to do a query across all of these databases. For instance, show me covariates in all patients who have mutations in TP53 across all of these cancers. And so this is another kind of thing that Phenopackets can be useful for. So I'll stop there and take questions.